that, I would like to one simple question. Vignesh, can you present us your PhD research?
Thought you'd never ask. Thank you. Okay, just give me a second. All right. Hey, hello everyone. Thanks for joining my talk. I'm Vignesh Ravichandran. I'm a final year PhD student working on designing smart wearable systems, and particularly my research over the years has looked into how we can look at both the human factors consideration along with the technical aspects of building a wearable. Yes, so this is kind of a rough overview of how my talk will go. So initially I'll kind of give you a quick introduction. After that we'll go into 3 studies which I've chosen, and then finally we'll have the conclusion. Okay. No, the mic's not working. Okay, sorry about that. Okay, so let's start first with the design of wearables. So wearable systems refer to any body-worn sensing systems that can measure physiology across the body. By its very nature, we can capture very different bodily parameters, and this gives us an insight into the overall health and well-being of an individual. So when we look at a clinical wearable system, what does it comprise of? So first we start with the signal that the body produces. So this can be the underlying physiological process, which is picked up by any type of sensor interface we can use. And then we design embedded hardware, which can pick up these signals, store it, or transmit it. And then finally, we have some computing layer on top, which can extract some insights from it, and finally present it in a form that has some real clinical utility. So when we look at wearables, I look at it through 2 lenses. First is the wearability, which is shown in the x-axis, where basically it's the amount of time that an end user is comfortable wearing this device and how much they'll tolerate it. So, and on the y-axis, we have the clinical fidelity of how good the signal we get from it would be. So in this sense, if we look at it, consumer wearables like the smartwatches and smart rings everyone uses is more in the bottom left corner, bottom right corner, where like it has high on usability, but not so much clinically insightful. But clinical devices like what you see in a doctor's office provide way more insights into the health, but unfortunately they're not tolerated for long duration. My research specifically looks at this particular top right corner where like we can, and looks into inquiring whether we can create systems that are both usable and well adopted, along with giving us clinically relevant insights. So let's go into a quick primer on, since most of my research focused on cardiac-based signals, so I thought it'll be a good primer to give on the sensing mechanism for the same. So for optical sensing, like what most of our smartwatches use, it uses an active measurement system by sending light into the body and looking at the reflected light to look at how much the blood volume change corresponds to it. So this ends up roughly how it works. And this gives us an idea on the overall blood volume change and then heart rate. The other approach most, especially clinical devices use, and some of the smartwatch when you hold with your other hand, is basically electrocardiogram. So electrocardiogram measures the electrical signals associated with the contraction and relaxation of different chambers of the heart. So basically the biggest peak we see is more due to the ventricular activity, which corresponds to this R wave, which is useful in calculating the heart rate. So now let's think about how we go from that to the form factors we could use for ECG. So clinically, outside of a standard resting ECG you get in a doctor's office, if there's an abnormal rhythm which doesn't manifest itself during the brief clinical visit, doctors would send you home typically with these kind of devices, which are Holter ECG monitors. They have this rather bulky monitoring device. Along with that, they also have these different electrodes attached to it. By its very nature, it's quite cumbersome and not well adopted by the user, even though it has higher signal quality. So after that, we have arm ECG, which basically gives us more continuous measurement potential, which is more well adjusted, and especially works better across all the genders. And on the flip side, it is much smaller of a signal, so it's more susceptible to noise. So let's go into my research objectives. My first objective is to see how we can quantify placement and especially form factor decisions we make with respect to how that affects the signal compared to a clinically accepted site. The second objective is to see that if we go with the suboptimal site, how do we recover the biosignal information from it? And third is to look at the interplay between the sensing, computing, and human factors across different clinical populations. So this is kind of how I roughly visualize those aspects. So I look at it as 3 pillars, all tugging on each other, and the ideal wearable would be somewhere in the middle, where it kind of satisfies the needs of all and would have clinically useful insights, but also be well adopted by the end user. So my research journey overall has been like a meandering river. Primary branch has been through the lens of opioid use disorder, and that's kind of will be the focus of this particular talk. But then I did do a lot of other work related to Parkinson's disease monitoring, especially movement sensing and user-based design, and also having some exploration on like impedance cardiogram, which for more cardiac care monitoring. So let's go now into the dissertation context specifically. As I mentioned, opioid use disorder is the primary focus of this research. So what are opioids? So opioids are a class of painkiller drugs, which basically, through its overprescription, has led to widespread misuse and addiction. People develop a high level of dependence, and over 1 in 58 people in the last year alone are known to have been suffering from this disorder. And this also leads to a good bit of human fatalities. Over one person dies every 12 minutes due to this, according to the latest CDC stats. So let's look at now how the opioids actually work and why they're so addictive. So opioids work on the pain receptor. So by directly binding to it, initially people start off by a prescribed dose, which over time they develop tolerance, and this leads them to kind of get a higher and higher dose, and before you know it, they're using street drugs like fentanyl or heroin. And getting off of opioids are just as hard as well. So there are a lot of severe withdrawal symptoms such as sweating and anxiety that initially starts if the person decides to walk the road to recovery, but then the peak withdrawal starts after. So some of the patients I've interacted with have described it as the worst flu you've had, along with the feeling of being hit by a car. And that's kind of the withdrawal symptoms that they have to put up with. And even if you get through that stage, the withdrawal symptoms still persist, especially days after, where like days or weeks and even years after, they're still having lingering comorbid mental disorders, along with intermittent cravings. So how do we provide care for this? So currently clinicians use a whole set of drugs, including methadone, which is a full opioid, but however it's quite controlled, so it needs a prescription and regular dosing by a clinician. And on the other end of the spectrum, you have naltrexone, or commonly known as Narcan. While it's wonderful in reversing an overdose, it has severe withdrawal symptoms, so it's not suitable for continuous titration of this particular drug. In middle, we have buprenorphine, which basically is a partial agonist, which means it doesn't give the high sensation, but it manages the cravings and withdrawal symptoms of the individuals. But still, getting that dosage is critically important. So the current way doctors go about it is they basically give clinical assessments, which look at subjective metrics along with the self-reported symptoms of the patient to basically titrate the dose based on standard clinical protocol. And if they don't get it right, their withdrawal can kind of persist, and this could eventually lead them to relapse and get off the wagon. So this brings me to my overall project vision. So my project vision is that can we use objective biomarkers from wearables to manage treatment of opioid use disorder? And basically looking further, basically it involves designing a wearable, extracting features from it, and then looking at how we can model the underlying physiology of a drug being used to hopefully enable dose management. So I mentioned objective biomarkers, so what are they for opioid use and withdrawal? So opioids work on the autonomic nervous system, and because it has the whole link to the different bodily mechanisms, it increases the aspects such as heart rate, skin conductance, so people are more sweaty as a result, and also they're more restless, which increases the body movement. So what kind of form factor would be accepted by this community now that we know that what are the signals we need to measure? So for this part, we worked with our with our collaborators in the assets lab at URI, who do a lot of human factors research, and they basically did semi-structured interview with over 14 people with lived experience or managing opioid use disorder. And we worked together to develop different form factor options, and basically we showed them for feedback. And basically we came up with diverse form factors which would both maximize the signal quality, but also we thought would also be higher on the usability end. So out of these form factors, we found that the armband was the most well adopted and preferred by the population. And the main thing that they were looking for in these wearables was their first priority was concealability. As you can imagine, with the stigma associated with this disorder, individuals are really skeptical of interacting with the medical system and do not want to be adversely persecuted for that. And that's something that they want with the armband, where it could be potentially worn under the clothing, so it wouldn't be visible to an outside person. And things like comfort and also ease of use is also important because they're already going through a lot, and we're expecting them to wear this device for multiple days. So this got us to our concept, which is the Minder upper armband. So we had initially conceptualized this, but the usability studies really did give us an idea that the armband is the right way to go. So this is a band that has different sensors, including the single-arm ECG electrodes, along with optical heart rate sensing, and skin conductance measurements, and temperature. And temperature is really important because that was a deciding factor between a wristwatch and an armband, because in the wrist, the temperature we measure is more closer to skin temperature, whereas in the upper arm, we get closer to the armpit, which gives us a more closer measurement of the upper arm temperature, armpit temperature. So I mentioned earlier that we need both single-arm ECG and optical heart rate. Why? So single-arm ECG and optical heart rate signal, such as the smartwatch technologies, has been known to be plagued with heart rate inaccuracy issues, especially with individuals with darker skin tone. And it's completely not functional in individuals with tattoos, which is quite an overlap with the population I work with. The other aspect is also the aspect of power consumption. So because it's an active measurement, we end up sending more current in for the measurement compared to a passive measurement like ECG. So this makes continuous recording require a larger battery and therefore a more bulkier device. The final aspect is more an opportunity than a specific con. By recording both ECG and PPG together, we can get an additional biomarker called pulse arrival time, which is known to be tracking blood pressure. And this is something that could be useful for modeling withdrawal as well. So this first brings me to my first study. So how do we go about actually designing the electrodes which would optimize, dry electrodes that optimize the collection of ECG? So before we go into that, there's a bit of basics we need to know about the skin electrode impedance. So skin electrode interface refers to the different tissue layers from the dermis to the stratum corneum, which is the outer layer of the skin, and any charge produced by the body has to travel all of these, and these can be modeled using resistors and capacitor networks. And we can see with the gel electrodes, or the standard ones you get in a doctor's office, those have an aqueous medium that covers those micropores in the skin. So because of that, it makes a perfect coupling and has only the resistive element. Whereas with the dry electrodes, there will be small minor gaps present, which lead to parasitic capacitances, which add up and increase the effective impedance. So for this particular design inquiry, my question was more centered on what type of electrode would be suitable. Assuming I keep the material, that is the silver polymer yarn, constant, can I vary these design aspects, such as stitch parameters, fill patterns, to make different electrodes, and how would that vary the skin electrode impedance? And one of those specifically was looking at particularly, one of those electrodes was a knit electrode, rest of those were the embroidered electrodes. So how did I go about making this? So to make the actual embroidered electrode, I used the ZSK technical embroidery machine, where I designed the pattern in the EPC Win software, and then I embroidered it on a base fabric, cut it out, attached an electrode tab. This is a rough demo of how that works. Okay, so now I have my embroidered electrodes. How do I make my knit electrodes? So for that, I use a standard knit conductive fabric, similar to what you get in for electromagnetic interference shielding. I attached it with fabric double tape and cut it out with a laser. Okay, so now that we have our electrode, I need a way to test it. So Human bioimpedance is continuously varying due to the underlying biological processes. So it doesn't suit as a perfect test bed for benchmarking electrodes. However, we can use these skin electrode phantoms, such as what I adopted from Goyal et al., which basically uses agar-agar with glycerol and then adds an amount of sodium chloride to mimic the skin electrical properties. So using these, I'm able to actually acquire and model the skin electrode properties. So this is a rough design of how that process worked. Once I had the solution, I pour it in these agar molds, and then basically once it cures with this buried electrode, I'm able to measure impedance. So this is kind of how my test protocol worked. I wanted to vary the contact pressure to see how the contact pressure interferes with the measurement, and also, so I varied it using slotted weights from 3 grams to 53 grams, and then I controlled exposure to sweat, so I would understand how perspiration affects, and then put it through controlled drying to see how post-exposure to sweat these electrode properties change. So this is a rough view of how my test protocol worked. I used an LCR meter, sweeping from 20 hertz to 5 kilohertz at 10 hertz intervals. And this whole process for a single electrode took like 10 seconds. Okay, so how did it vary? So this is kind of a rough view of how the electrode impedance properties varied across the different tests. So you can see for the different electrodes, especially the light blue color, which is the knit electrode, it's much closer to the black one, which is the gel electrode, which was interesting on its own. But then the interesting stuff happens when we expose it to sweat. So post-sweat exposure, you can see that the impedance level almost halves. And that persisted trend also changed when we put it to the controlled drying process. So this was in a conditioning chamber. So we can see that there's an increase by nearly twice of the baseline measurement post-exposure to sweat after controlled drying. So what did I learn from this? What I learned primarily was that my engineering mentality was like more conductive material equals lower impedance, right? But no, geometry and structure plays a divisive role. So knit structure in general had the best performance of all the electrodes under most of these cases. And of all the embroidered electrode, this particular crossbowl pattern, which has this kind of star structure, hub and spoke structure, seemed to reduce the impedance the most, because I suspect it has something to do with the lower path of conductance from the outside to the inside. And sweat also seems to have a more short-term and long-term effect on these performance of electrodes. So these are some of the references. And on to my next study, now that I have my electrodes, how do I measure arm ECG? And also, how is that, what kind of bioinstrumentation do I need to achieve this? So this is where some of those bad parts of arm ECG comes around. One, it has a much smaller signal amplitude. It's in microvolts compared to the millivolts for regular chest ECG. And there's significant motion due to muscle activity, which affects these arm ECG signals. And also, there are factors such as arm circumference, and the orientation of the electrodes also affect the performance. So this requires a special type of instrumentation, and also we need to validate it against the standard chest ECG configuration, so we know what's the trade-off. So this is kind of a rough view of 3 years of my research journey in a summary. So I started from a benchtop hardware and then shrinked it down over time with my team into the smallest form factor you see there. So this particular embedded system I came up with uses the MAX 30001G, which uses an 18-bit measurement for electrocardiogram. And then apart from that, I used a Raspberry Pico RP2040 microcontroller, which is a dual core controller. So I could sample the sensors through semaphore process and then store that onto an SD at the same time without it blocking or affecting the sampling rate in any way. And finally, it stores those data into an SD card. So on to the next part. So now I need to actually make the armband, right? So I took the knit electrodes from the first study, and basically I was able to use that, and I wanted to evaluate if a standard flexible PCB substrate would potentially work out. It's quicker to manufacture, more easier. So I evaluated a gold-coated flexible PCB electrode and kind of evaluated both of them by repurposing BP armband cuff, and we added a measurement tape and Velcro so that we can space the electrodes evenly across different individuals. So how did my protocol look like? So we used an IRB-approved protocol, which looked at the arm ECG signal with reference to the chest ECG signal and EMG while the person performed different movements. The movements particularly included movements which include the biceps, like the elbow flexion extension, and abduction, adduction, which uses the shoulder muscles, along with walking. And all of this data was collected using the standard BioPack MP160 system, so this is kind of how the setup looks like. So how does that actually affect the signal? So this particular scaleogram plot shows for one of the activities, which is the elbow flexion extension. And we can see that for the most part, the signal is clean. The chest ECG is obviously stronger, but we can see that the arm ECG does have the same cardiac beat information that the chest ECG does have. But once we have the person do a movement, as evidenced by the electromyogram or muscle activation, we can see that it's completely buried with the chest ECG, with the arm ECG. And even the chest ECG has some of those artifacts, but still the ECG part is still visible. So to get some more quantitative numbers on how this is affecting, I put together a signal processing pipeline, which involved these different signals put through a fifth order Butterworth bandpass filter between 0.5 to 45 hertz, which is the standard band for Holter ECG. And then I extracted the power spectral density and cross spectral density. And using that, I was able to compute the mean square error along with the EMG interference information and the band energy of 5 to 15 hertz, which is where the cardiac beat is at. So looking at the numbers, so this is, in this case, basically we can see that the mean square error is much lower for the silver knit electrodes compared to the flexible PCB electrodes. And the cross-spectral density, which looks at how much of the muscle activity is present in these particular electrodes, that also was much lower with the silver knit electrode. So now that I have that, I wanted to then test it with our custom hardware we made. So I evaluated it with a synchronized data collection protocol with my BioPack system, which basically collects the arm ECG and lead 1 ECG along with my custom hardware, which also seems to give a similar signal quality. So what did I learn from this? So first, I noticed that the bicep activation was adding way more muscle artifact contamination, but walking by far was the worst, especially with those rigid gold electrodes. Then the arm ECG and my custom hardware is capable of picking up these beats, especially for heartbeat sensing, and the textile electrodes continue to be better, so we continue on with it. And finally, the noise requires us to come up with special strategies to solve it. So this is kind of a journey of how the armbands themselves varied over time. I have one sample piece here. You can see it after. So on to the next part, which is kind of focusing on what we do with this data once we have these biosignals acquired. So this particular aspect was done in collaboration with UMass Chan Medical. So this represents a paper that's still under review in the Journal of Biomedical Health Informatics. So to understand this, we need to look at through lens of two core aspects which are fundamental to this particular research. So pharmacokinetics is the process by which scientists model how a drug is processed by the body, versus pharmacodynamics looks at what the drugs actually do to the body. So in case of pharmacokinetics, we can look at it through the lens of, based on how the drug was ingested or injected, the amount of absorption, distribution, and how it's metabolized and excreted out of the system vary. And these vary by the drug to drug and could vary across individuals too. And pharmacodynamics, particularly, as I mentioned earlier, in case of the opioids, is going to affect the autonomic nervous system. And because of that, we can model it by metrics such as heart rate variability and how the blood pressure also varies. So how does they currently study pharmacokinetics of a given drug? So when a drug is being produced, they do clinical studies at different concentrations to understand how the dose, how the body is metabolizing it. And that's done using the blood plasma concentration of a given drug. And they get these measurements at fixed interval. And basically, this particular aspect can be modeled using Bateman exponential functions. So this basically, in this particular equation, you can see that the drug concentration, bioavailability, and dose, and volume of distribution are more related to how the drug is packaged. But the absorption and elimination rate are aspects that are more individual based on the person's biology. So how does the research community currently assess the opioid use disorder state modeling? So some of the prior studies, such as Gulapi et al., had looked into basically using signals like electrodermal activity, temperature, and motion to basically try to predict the medication intake time. The other research approach that basically Kuhlmann et al. Had done was to add the pulse plethysmogram signals to it to basically distinguish between a binary class of on medication versus off medication. And finally, more recently, Cho et al. Had shown that they can use seismocardiogram along with electrocardiogram to try to regress towards the withdrawal score. There are still some open questions, however. So what kind of features are actually useful in distinguishing the pre and post medication effect? And also, can we capture the feature trajectory as it changes over time, especially as the medication is taking effect? And also, how can these multimodal measurements be fused together when we make these decisions? So for this is a kind of rough visual of how these different approaches are taking and how I want to pursue that. So one of the ways that I presented earlier was a binary classification or regression. And the way I want to take is to more look at the part where most people throw off, which is the process of absorption of the drug, to see how that's actually changing by the individuals. So to do this, we worked on a clinical study protocol with our partners at UMass Chan Medical. And basically this involved participants coming in who were in buprenorphine maintenance therapy, which means they're on a stable dose. And basically they come in the first thing in the morning, and we get a baseline measurement, and we get a dose intake cow score, which measures the withdrawal intensity through subjective markers. And then the clinicians observe them actually taking the dose, and all while they're put through our sensing protocol. And finally, they're recorded for about 5 hours in total. And after the dose was taken, after 90 minutes, they give the cow score again to get how the post metrics look. So the particular data set slice I'm using is for 15 participants, and they have a wide range of the dose medication they were on. And also, you can notice that the cow score is comparatively low. So cows is a score that varies from 0 to 48, where 0 is low withdrawal and 48 is peak withdrawal. And you can see that even pre-post is pretty similar. And for this particular data set, I used the clinical gold standard BioPAC device because I wanted a more high resolution signal first to model this. So we acquired the lead 1 chest ECG along with the fingertip PPG, along with noticing the dose timing and what the cow score is. So how do I now use this data? So for modeling this actual underlying physiology, I first put it through a pre-processing pipeline that uses the BioPAC data along with a bandpass filter, which is standard for both signals. And then I acquired 11-dimensional features for the signal quality indexes, which I kind of discussed a bit earlier. And then I retain the clean and motion artifact and reject the motion artifact windows. And based on this, I'm able to now then extract the clinically relevant parameters such as our peaks and the peak and valley of the optical pulse signal. And based on that, I'm able to extract these metrics such as RR interval, which look at the spacing between the beats, and then rise time of the PPG, which looks at the systolic upstroke, and then pulse width at 50% of the amplitude. And by combining ECG and PPG, which are synchronized by the BioPAC, I'm able to get a metric which is pulse arrival time. So this is kind of a rough view of how the pre-processing pipeline works. So I used this, I made this particular graphical user interface, which lets me upload the BioPAC and the label files, and then I'm able to cluster these based on the signal features that I mentioned before. And the ones in yellow are the cases where it's artifact, and because it's 5-hour protocol, patients will go on a break. So those would kind of help me eliminate those windows which are not useful. So this is kind of an example of like good windows in the top, and bottom is one of the windows or both of the signal streams are corrupted. So this is kind of the beat detection and the fiducial point detection and the corresponding feature metrics that accompany it. Okay, so by doing all this clustering process, I'm able to retrieve about 85.7% roughly percentage of all the data. And the average recording period was around 5.6 hours, so that's a lot of data to play around with. So the first question I had was to see how these different biomarkers respond to the dose states. So for this, basically, I wanted to look at the pre and post changes. And for this, I used the linear mixed models, which help me basically identify the significance of each feature. And we can see that all of the features, which I described earlier, were significant, and having an a larger value pre and post. And okay, so now that I have these features, what do I do with it? So basically, I take these features, and I need to fuse it. And for the fusion, I use a PCA and Kalman model, which basically combines the 4-dimensional features down into 1 dimension for each timestamp of 1 minute. And then basically, for each of those, I'm able to then put it through an autonomic, I call it the autonomic state indicator, and I smooth it using the Kalman smoother model, which basically lets me fill up those gaps. If there are any, or if there are any artifact windows that got rejected, it helps me adapt to those. And after that, I put it through the PELT model to detect the onset of the dose intake, and hopefully detect when that dose started having an effect. And finally, I then compare it with the population level pharmacokinetic model, which is derived from prior clinical studies, and then compare the shapes of the response that I measure, which is the autonomic state indicator, and the population PK model. And then I also calculate the feature trajectory using the p-hate model. And using all this, I come up with a rough phenotype of the responders. So this is not clinically measurable, but something that helps me understand which of these patients' autonomic state indicator match the population PK model. So based on that, I found in my data set, I was able to find 8 strong responders, 4 weak responders, and 3 people who I couldn't detect any response. So this is kind of how that ASI metric looks. So you can see the dotted line here. So that's the Bateman model, which were derived from the pharmacokinetic models from the research community. And then we can basically see that the purple color one plot is the one showing the strong responders. And we can see that there is a bit of overlap there. And whereas for the individual metrics, even though they share a similar increase, it's not as pronounced as the autonomic state indicator that I derived. And the feature onset classifications also show that. So for the fusion condition, we detect way more onsets with the sensor fusion compared to the individual feature metrics. Okay, so what did I learn from this? So the big takeaways from this was that for the baffling part for me was that even the cases where the cow score was flat, which I would expect to see a similar response in the autonomic state indicator, my model was able to see a shift in the biosignal. And in cases where they were subclinical by standards, it does seem to be that those changes are detected. This kind of now makes sense, though, now looking back and consulting with the clinical partners, because the population I worked with are the ones in the maintenance cohort. And the cow score is primarily made for dosing and adjusting that, so it's made more for the people in acute withdrawal symptoms. So this shows that feasibly I could detect this even in cases where people are in the longer term care. So other aspect which I found interesting was that the drug onset time is quite variable across individuals. And finally, fusing the ECG and PPG features seems to give us more richer context on their actual recovery compared to the individual signals. So now that I have all this, how do I build on this insight to go to the next steps? As I mentioned earlier, this particular data set was more analyzing the BioPack data set, which used the standard configuration, but then that's not really feasible for the environment we need to be in, which is the initiation part, which might happen in an inpatient clinic, rehab facility, or the patient's home. And we can't exactly send a multi-$1,000 research grade system to all these places. So that's where the armband really comes in. So because of the cost associated with it, we're able to actually send this out potentially to get these markers. But with that comes a lot of challenges, as Uncle Ben said. So basically, the phase where we need to actually measure is the case where the signal is the worst, because these people are undergoing some of the most severe withdrawal, and we expect to see a lot more tremors and motion artifacts, which require us to make some improvements in our signal pipeline if we need to get the armband to work here. So for making this work, since all of these analyses rely on beat detection, we need to strengthen the R-peak detection capability of the ARM ECG. And also, we need to be aware which windows are actually artifact and which are the windows where there's a good bit of signal there. And also, we need to enhance the usability of this device so it actually survives unsupervised use. So this kind of neatly segues into another topic, which is more on basically developing a custom beat detector for ARM ECG. So for doing this, basically, I won't have time to go too much into it, but if anyone has to know, I can go over a bit later. But basically, I developed a custom unit model, one-dimensional unit model, which basically uses the distance transform, and I basically adapted to the lack of the ARM ECG datasets that are there publicly by transforming the large corpuses of chest ECG signals we have into 767 days worth of pseudo ARM ECG. By doing this pre-training regimen, I was able to zero-shot detect on actual ARM ECG signals, and then I was able to run the model light enough on a Raspberry Pi 5 in under 24 milliseconds. So this is a rough demo of how that works. So in this case, you can see that the beats are all pretty visible. So I use a lightweight standard beat detector, and then I use a standard beat detector. Sorry. Yeah. And then I use a standard, our custom RPNet detector when there's motion noise present, like what you see here. And even in those circumstances, my detector is able to detect those specific R peaks. So these are some of the references for the last two studies. And then now let's put these all together. So from my first study, I learned that the bench performance doesn't always translate to the real body. And with the ARM ECG signal, it was kind of similar. I first tested with benchtop hardware, and with my hands as still as possible, and then the moment I added some amount of noise, completely everything fell apart. So second question was more, objective was more looking into, can we use computational methods to recover the lost biosignals? Yes, but within a certain limit. So with ARM ECG particularly, I was able to observe that I'm able to retrieve the certain slice where there's some moderate amount of motion corruption, which normal beat detectors won't detect, but it can't make data out of nothing. So that's still a constraint. Final objective was more on basically, can we use this co-design process, does it do anything? And for this, I developed more of a nuanced take. I was trying to perfect all three of these pillars together, but that turned out to be optimizing none in the end. But what I did take away from this journey was that the human factors element is often the most binding constraint because that defines whether the device is worn or not. We can make the most fancy gadget we make, but then if it's not worn, it's not getting any signal. So in my view, I think sensing and the computational methods can offset some of those trade-offs we get due to human factors, and that's something that I think we can all build on. So looking at where we can go next with this. So primarily with sensing, I think improving the coatings on these electrodes so that we can manage the sweat-related influence and also increase the skin electrode impedance. That's something that's pursuable. And also making it more personalized manufacturing, like my colleague Shubham had made earlier presentation on. The other aspect is more on computational methods. So this is where having synchronization, instead of going where the variable is, making the variable pick the signal where there's none, we go where the signal is and kind of maybe synchronize. And finally, with the human factors element, as I mentioned, these individuals have a lot of skepticism with the medical system and governments. So I think on-device AI and edge inference are going to be key to getting inference in a way they trust and actually make them trust and use these kind of devices. All right, so I said a bit about distributed sensing. So this is kind of how I view. So instead of going where the signals are, what if we can make these variables talk to each other and actually make it stronger together? And this is kind of roughly what I mean. We are seeing a lot of these form factors such as smart rings, like I wear both my smart ring and my smartwatch, but they all can measure physiology by their own. But maybe if we can get them to talk to each other, like what I did with my pulse arrival time, we can maybe unlock more clinically valuable insights than each one would on their own. So thank you all. So this is kind of the list of my publications that I have as a first author. And I had the privilege of working with several other researchers in this community and our lab, and I was able to publish all these other works too. And my work wouldn't have been possible without the support of the National Institute of Health, which supported the Minder opioid use study. And then I also went through the NSF I-Core program, and I was a P2B fellow last year with the Rise Up Foundation. And also I was an intern at Bell Labs where I did one of my research for my dissertation. And also the humans that made it all happen. I think it was a long journey. So true story, I knew Kunal from my high school days. I was sending him emails to send me full text of his dissertation. And we stayed in touch all these years. And I can't see me doing the PhD with anyone else. And same with Dhaval. I think I learned a lot from them. And from my lab people, I think they kept me sane throughout this journey. And my particular community, I think RIP Grad Village, I think we all missed that. But it was really tight-knit community, and we were all there to support each other. And best of all was the family part. So I think this year was a special year. I also got married. And yeah, it was kind of a transformational moment. And for outreach stuff, I did do a lot of, I had the privilege of doing a lot of outreach events, teaching young people, and also presenting my work in the community and winning some awards at sometimes. So thank you all for joining me, and if there's any questions.
Thank you, Viggy. We are short on time and fabulous presentation, and also thank you for bringing all the, the work that you have done beyond research as well. Yes, Viggy is right. He was in his 10th standard when he contacted me. I was postdoc, by the way, at that time, and I thought I could have a PhD student like this in the future, and it really happened. So thank you, Viggy, for joining our lab. Yes, it was several years of work. You have seen it just now. And what we will do right now is that we will take some questions, around 3 or 4 from audience. And after that, we will kind of ask audience to leave. So, questions from audience. Yes, Alex?
So, I believe it was your second or third study, you were discussing some of the signals you reported from, and you briefly mentioned an EDA signal.
Mm-hmm.
I wasn't quite certain what that was. I haven't heard of it before. Can you go into a little bit
Sure.
what it is?
For sure.
Okay, the question is about the EDA and
Yes.
defining it.
Okay, so basically that kind of segues me into something I found. So electrodermal activity is a signal that measures the sympathetic activation. So that's more response to the stress and or fight or flight response. And that basically is a measurement of the pores in the skin, basically the sweat glands activating. And that's a very specific type of sweat gland, which we kind of learned the hard way. Let me see if I can go into that. Yep. So for measuring that, basically it measures as these spikes. It has a slow level rising peak, which is called SCL. That's the skin conductance level. And there's skin conductance response, which is more of an AC information, like these spikes we see here. So we learned it the hard way that in the upper arm band, we couldn't actually measure that signal. So even though that was one of the goals we had, so we had to run wires all the way to the finger. So commonly we would have seen these technologies either in lie detector test. So that's like a common application of this particular signal. And so this is also where I think the body sensor network aspect really comes in, because I think instead of forcing technologies to measure that where there's no signal there, we could kind of make it like these ring form factors, which basically can measure this sympathetic activation. And it is a slow moving signal, and uniquely, whereas most of heart rate variability signals capture both the parasympathetic and sympathetic activation, with EDA particularly, it only reacts to the sympathetic activation. So that makes it quite a useful signal from if we're doing autonomic nervous system based studies. I hope that answered your question.
Yes, very, very well. I wasn't familiar with this term, and it seems that the EDA is tracking the sweat gland pore size that can then lead to detecting the amount of perspiration that a person is forming at a particular time, which, as you learned earlier, directly ties into the impedance of the ECG monitors themselves. So you have a measurement for how much they're sweating, and that controls for the impedance and the signal quality of their heart rate monitors later down the line. So
Yes.
a more holistic view.
Yes, we do get a more holistic view with this. Yeah, sure.
Pinaki from Zoom asks, did you notice any differences across genders in the findings?
So I hadn't had the chance to look at it with the PharmaDT response. It did seem most of the participants had a consistent.
Please repeat the question.
Sorry, just for the audience. So Meenakshi had asked if there's any difference in the findings across the genders. Particularly with the arm ECG one, we hadn't noticed much of a difference. One difference was, I think, female genders had used more of the hydration, like basically moisturizers. So in fact, it was more easier to get signals from them compared to us guys who have more drier skin most of the times. So apart from that, there wasn't much of a difference. Even though the PharmaDT study was pretty similar across the board. Hope that answered the question. Any other?
For your, I saw that you used silver electrode and silver thread, and you used different types of structures. Now, if anyone wants to design this kind of electrode for ECG or for ECG, so what would be your suggestions in terms of design, placement?
Sure. I think, so the question from Ramon was basically, since I'm using the silver-based electrodes, especially textile electrode, what would be my guidelines and suggestions for people who are designing wearables for ECG? That's right. So I think the main aspect is basically contact pressure. I think contact pressure makes a really big difference in the quality of signals we get, and fit is a very big part too. So because the skin electrode impedance, to break that, you need to kind of couple it through those micropores. Coatings is another excellent option, which I hadn't pursued, but I wish I had. Basically, there are a lot of work on silicone-based coatings, which basically can kind of spread out that gap. And there are also researchers working on semi-dry electrodes, which basically perpetually keeps coating the surface with some gel. So it's mostly dry for the most part, but there's like a small element in it. So I think that's kind of my biggest takeaways, I would say. Analyze both the structure and the impedance characteristics. Yes, Lloyd.
So, as you design this device, I'm trying to say, like, what kind of clinical decisions you think could be made using the data that you can get?
Okay. Lloyd's question was more on what kind of decisions can we make clinically with this kind of wearable device, if that's right. So I think particularly my research was more focused on the opioid use disorder population. So I think withdrawal is the direct step to go. But I think an extension of this would be stress management. Because I am looking at the autonomic nervous system processes, I think kind of having a continuous monitoring of ECG and PPG is something that could help people diagnose hypertension much earlier and kind of manage interventions for hypertension. So that's kind of how I see this going. Yeah, Jip.
Unlimited time and funding, what is the problem that you want to work on?
Okay, that's the funnest question. If I had unlimited, Jim was asking me if I have unlimited funding and time, what would I be doing? I think personalized manufacturing. I think that's been a really big dream of mine to make happen over the years. I think developing technologies that actually make e-textiles more scalable and address these clinical needs, I think that's something that I'm really passionate about, and at least something I look forward to doing in the future, if not now. Last one, sure.
Very, very interesting. Learned a lot of things here. I think you have a great application in neonatal field, because neonatal abstinence syndrome is still done with the scoring, and we still do not have any kind of mechanism the way you have done it for adults.
I see.
So if you can translate it, it can be a very big, especially neonatal abstinence syndrome is very big in Europe.
Abstinence.
Where the parents are, mothers are on drugs and the babies are withdrawing.
I see.
And you have
Okay.
to keep the babies in nursery for 4 to 5 weeks and score it,
I see.
which is not as easy as an adult. So it's an amazing translation which can happen for you commercially.
For sure. Thank you so much. I'll keep that in mind.
Thank you, thank you. Okay, everyone, thank you, audience. Thank you very much. And we'll keep it in a closed door now. And
Yeah.
there is some coffee and everything if you like to enjoy that outside.